Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 2:01 a.m. Eastern Time. Quick note. Super cool water doesn't have to freeze until minus 48 degrees Celsius, minus 55 degrees Fahrenheit. Science Daily, November 23, 2011. We drink water, bathe in it, and we are made mostly of water. Yet the common substance poses major mysteries. Now University of Utah chemists may have solved one enigma by showing how cold water can get before it absolutely must freeze. 48 degrees below 0 Celsius, minus 55 degrees Fahrenheit. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 2:02 a.m. Eastern Time. Quick note. That's 48 degrees Celsius, 87 degrees Fahrenheit, colder than what most people consider the freezing point of water, namely 0 degrees Celsius, 32 degrees Fahrenheit. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 2:04 a.m. Eastern Time. Quick note. Super cold liquid water must become ice at minus 48 degrees Celsius, minus 55 degrees Fahrenheit, not just because of the extreme cold, but because the molecular structure of water changes physically to form tetrahedral shapes, with each water molecule loosely bonded to four others, according to the new study. And good morning, it's Dr. Will, CBMD, PhD. It's Tuesday, November 29, 2011, 2:05 a.m. Eastern Time. Quick note. The findings suggest a structural change from liquid to intermediate ice explains the mystery of what determines the temperature at which water is going to freeze. An assistant professor of University of Utah, senior author of the study, published the November 24th issue of the Journal of Nature. And good morning, it's Dr. Will, CBMD, PhD. It's Tuesday, November 29, 2011, 2:06 a.m. Eastern Time. Quick note. This intermediate ice has a structure between the full structure of ice and structure of liquid. We are solving a very old puzzle of what is going on deeply super cool water. And good morning, it's Dr. Will, CBMD, PhD. It's Tuesday, November 29, 2011, 2:07 a.m. Eastern Time. Quick note. However, in the strange and wacky world of water, tiny amounts of liquid water theoretically still might be present even as temperatures plunge below minus 48 degrees Celsius, minus 55 degrees Fahrenheit, and almost all the water has turned solid, either into crystalline ice or amorphous water glass. But any remaining liquid water can survive only incredibly short time, too short for the liquid's properties to be detected or measured. And good morning, it's Dr. Will, CBMD, PhD. It's Tuesday, November 29, 2011, 2:07 a.m. Eastern Time. Quick note. How and what temperature water must freeze has more than just gee whiz appeal. Atmospheric scientists studying global warming want to know at what temperatures and rates water freezes and crystallizes into ice. And good morning, it's Dr. Will, CBMD, PhD. It's Tuesday, November 29, 2011, 2:08 a.m. Eastern Time. Quick note. We need not to predict how much water in the atmosphere is in a liquid state or crystal state, which relates to how much solar radiation is absorbed by atmospheric water and ice. This is important for predictions of global climate. Good morning, it's Dr. Will, CBMD, PhD. It's Tuesday, November 29, 2011, 2:09 a.m. Eastern Time. Quick note. A strange substance. Liquid water is a network of water molecules, each with two hydrogen atoms and one hydrogen atom, held loosely together by what is called hydrogen bonding, which is somewhat like static cling. Depending on its temperature and pressure, water ice has 16 different crystalline forms, in which water molecules cling to each other with hydrogen bonds. And good morning, it's Dr. Will, CBMD, PhD. It's Tuesday, November 29, 2011, 2:10 a.m. Eastern Time. Quick note. What makes water so strange is that the way liquid water behaves is completely different from other liquids. Ice floats on water while most solids sink into their liquid forms because they are denser than the liquids. And good morning, it's Dr. Will, CBMD, PhD. It's Tuesday, November 29, 2011, 2:11 a.m. Eastern Time. Quick note. Water's density changes with temperature and is most dense at 4 degrees Celsius, 39 degrees Fahrenheit. That's why fish survive with their ice covering a pond by swimming in the water. Denser water at the bottom of the pond. And good morning, it's Dr. Will, CBMD, PhD. It's Tuesday, November 29, 2011, 2:12 a.m. Eastern Time. Quick note. But the property of water that is most fascinating is that you can cool it down well below 32 degrees Fahrenheit, 0 Celsius, and it still remains at liquid. And good morning, it's Dr. Will, CBMD, PhD. It's Tuesday, November 29, 2011, 2:14 a.m. Eastern Time. Quick note. Liquid water as cold as minus 40 degrees Celsius, minus 40 degrees Fahrenheit, has been found in clouds. Scientists have done experiments showing liquid water can exist at least down to minus 41 degrees Celsius, minus 42 degrees Fahrenheit. Why doesn't water necessarily freeze at 0 degrees Celsius, 32 degrees Fahrenheit like we were taught in school? And good morning, it's Dr. Will, CBMD, PhD. It's Tuesday, November 29, 2011, 2:17 a.m. Eastern Time. Quick note. If you have liquid water and you have to form ice, then you have to first form a small nucleus or seed of ice from the liquid. The liquid has to give birth to ice. For rain, you have to make liquid from vapor. Here you have to make crystal ice from liquid. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 2:18 a.m. Eastern Time. Quick note. Under which conditions do the nuclei form and are large enough to grow? And what is the size of this critical nucleus? Good morning, it's Dr. Will, CBMD, PhD. It's Tuesday, November 29, 2011, 2:19 a.m. Eastern Time. Note that when the cool down water, its structure becomes closer to the structure of ice, which is why the density goes down. And this should be reflected in an increased crystallization rate. Good morning, it's Dr. Will, CBMD, PhD. It's Tuesday, November 29, 2011, 2:20 a.m. Eastern Time. Note super cold water has been measured down to about minus 41 degrees Celsius, minus 42 degrees Fahrenheit, which is the homogeneous nucleation temperature, the lowest temperature at which the ice crystallization rate can be measured, as water is freezing. Below this temperature, ice is crystallizing too fast for any property of the remaining liquid to be measured. Good morning, it's Dr. Will, CBMD, PhD. It's Tuesday, November 29, 2011, 2:21 a.m. Eastern Time. Note computers provide a microscopic view that simulates that experiment cannot provide. This behavior of supercooled water was simulated and also modeled using real data. Good morning, it's Dr. Will, CBMD, PhD. It's Tuesday, November 29, 2011, 2:22 a.m. Eastern Time. Note previous computer simulations and modeling were too slow and had to last long enough for the freezing process to occur, and it was necessary to simulate thousands of nucleation events to make valid conclusions. Good morning, it's Dr. Will, CBMD, PhD. It's Tuesday, November 29, 2011, 2:23 a.m. Eastern Time. Note device to computer model that is 200 times faster than predecessors. The model simplified the number crunching by considering each 3-atom water molecule to be a single particle similar to a silicon atom and capable of sticking together with hydrogen bonding. Good morning, it's Dr. Will, CBMD, PhD. It's Tuesday, November 29, 2011, 2:24 a.m. Eastern Time. Note even so, it took thousands of hours of computer time to simulate the behavior of 32,768 water molecules much smaller than a tiny drop of water to determine how the heat capacity, density, and compressibility of water changes as it is supercooled, and to simulate how fast ice crystallizes within a batch of 4,000 water molecules. And good morning, it's Dr. Will, CBMD, PhD. It's Tuesday, November 29, 2011, 2:25 a.m. Eastern Time. Note the birth of ice, the computers helped determine how cold water can get before it reaches its theoretical maximum crystallization rate. It must freeze. The answer minus 48 degrees Celsius, minus 55 degrees Fahrenheit. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 2:26 a.m. Eastern Time. Note the computers also showed that as water approaches minus 48 degrees Celsius, minus 55 degrees Fahrenheit, there is a sharp increase in the proportion of water molecules attached to 4 others to form tetrahedrons. The water is transforming to something else, and this something else is very close to ice. And she calls it intermediate ice. And good morning, it's Dr. Will, CBMD, PhD. It's Tuesday, November 29, 2011, 2:28 a.m. Eastern Time. Note if a microscopic droplet of water is cooled very fast, it forms what is called a glass, low-density amorphous ice, in which all tetrahedrons of water molecules are not lined up to form perfected crystals. Instead, low-density ice is amorphous like window glass. The study found that as many as 1/4 of the molecules in the amorphous water glass are organized either as intermediate ice or as tiny ice crystals. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 2:30 a.m. Eastern Time. Note when water approaches minus 48 degrees Celsius, minus 55 degrees Fahrenheit, there is an unusual decrease in density and unusual increase in heat capacity, which goes up instead of down, and compressibility. Water gets easier to compress as it gets colder. Unlike most liquids, these unusual thermodynamics coincide with liquid water changing to the tetrahedron structure. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 2:31 a.m. Eastern Time. Note the change in structure of water controls the rate at which ice forms. We show both thermodynamics of water and crystallization rate are controlled by the change in structure of liquid water that approaches the structure of ice. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 2:43 a.m. Eastern Time. Note monarch butterfly genome sequence. Science Daily, November 23, 2011. Each fall, millions of monarch butterflies from across the eastern United States use a time-compensated sun compass to direct their navigation south, traveling up to 2,000 miles to an overwintering site in a specific grove of fire trees in central Mexico. Scientists have long been fascinated by the biological mechanisms that allow successive generations of these delicate creatures to travel such long distances to a small region, roughly 300 square miles in size. And good morning, it's Dr. Will, CBMD, PhD. This Tuesday, November 29, 2011, 2:44 a.m. Eastern Time. Note to unlock the genetic and regulatory elements important for this remarkable journey, neurobiologists UMass Medical School are the first to sequence and analyze the monarch butterfly genome. And good morning, it's Dr. Will, CBMD, PhD. This Tuesday, November 29, 2011, 2:45 a.m. Eastern Time. Note migratory monarchs are at least 2 generations removed from those that made the journey the previous fall. Professor and Chair of Neurobiology, senior author of the study, they have never been to the overwintering sites before and have no relatives to follow on their way. There must be a genetic program underlying the butterfly's migratory behavior. We want to know what that program is and how it works. Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 2:54 a.m. Eastern Time. Understanding the relationship between genes, behavior, and physiological adaptations in monarchs may also lead to new insights into similar connections in humans. Circadian clocks, for instance, are a crucial component in the complex time-compensated sun compass system governing a monarch's ability to navigate long distances, and are now understood to play a pivotal role in human biology. Temporal variations in hormone levels, pharmacokinetics, and disease processes, such as the increased incidence of heart attacks in the early morning, reveal the prominent influence of the circadian clock on human physiology. Understanding the molecular mechanisms of the circadian clock has already helped reveal how the clock gene mutations contribute to disorders of the timing of sleep, and new insights could illuminate how clock gene mutations contribute to disease such as the major depression and seasonal affective disorder. Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 3:01 a.m. Eastern Time. Identifying genes involved in visual input and central processing by the sun compass. A full repertoire of molecular components of the monarch circadian clock, all members of the juvenile hormone biosynthetic pathway, whose regulation is critical for successful migration and which shows an unexpected regulation pattern. Additional molecular signature of oriented flight behavior, monarch-specific expansion of odorant receptors potentially important for long-distance migration, and a variant of the sodium-potassium pump that underlies a valuable chemical defense mechanism to fend off predators during the migration. Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 3:01 a.m. Eastern Time. In terms of fundamental brain processing, those involved in the navigation mechanism used for long distance migration have been difficult to decipher. Dissecting the genetic basis of long-distance migration in the monarch may help us understand these mechanisms not only in monarchs, but more generally in other migrants, including migratory birds and sea turtles. Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 3:05 a.m. Eastern Time. Insect cyborgs may become first responders to search and monitor hazardous places. Science dealing November 23, 2011 research conducted at the University of Michigan College of Engineering may lead to the use of insects to monitor hazardous situations before sending in humans. Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 3:06 a.m. Eastern Time. Through energy scavenging, we could potentially power cameras, microphones, and other sensors and communication equipment that an insect could carry abroad a tiny backpack. We could then send these bugs into dangerous or enclosed environments where we would not want humans to go. Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 3:06 a.m. Eastern Time. The principal idea is to harvest the insect's biological energy from either its body heat or movements. The device converts the kinetic energy from wing movements of the insect into electricity, thus prolonging the battery life. The battery can be used to power small sensors implanted on the insect, such as a small camera, microphone, or a gas sensor, in order to gather vital information from hazardous environments. Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 3:08 a.m. Eastern Time. A spiral piezoelectric generator was designed to maximize the power output by employing a compliant structure in a limited area. The technology developed to fabricate this prototype includes a process to machine high aspect ratio devices from bulk piezoelectric substances with a minimum damage to the material using a femto laser. Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 3:31 a.m. Eastern Time. First dogs came from East Asia, genetic study confirms. Science Daily, November 23, 2011. Researchers at the Sweden KTH Royal Institute of Technology say they have found further proof that the wolf ancestors of today's domesticated dogs can be traced to southern East Asia. Findings that run counter the theories placing the cradle of the canine line in the Middle East. Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 3:33 a.m. Eastern Time. Data on genetics, morphology, and behavior show clearly that dogs are descended from wolves. But there's never been scientific consensus on where the domestication process began. Our analysis of Y chromosomal DNA now confirms that wolves were first domesticated in Asia South. We call it the ASY region in southern and China Southeast Asia. Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 3:35 a.m. Eastern Time. Approximately half of the gene pool was universally shared everywhere in the world. while only the ASY region had the entire range of genetic diversity. This shows that gene pools in all other regions of the world most probably originate from the ASY region. And good morning, Dr. Will, CBMD PhD. This Tuesday, November 29, 2011, 3:36 a.m. Eastern Time. Quick note. Since other studies have indicated that wolves were domesticated in the Middle East, we wanted to be sure nothing had been missed. We find no signs whatsoever that dogs originated there. In their studies, the researchers also found minor genetic contributions from crossbreeding between dogs and wolves in other geographical regions, including the Middle East. And good morning, Dr. Will, CBMD PhD. It is Tuesday, November 29, 2011, 3:37 a.m. Eastern Time. Quick note. In their studies, the researchers also found minor genetic contributions from crossbreeding between dogs and wolves in other geographical regions, including the Middle East. And good morning, Dr. Will, CBMD PhD. This Tuesday, November 29, 2011, 3:55 a.m. Eastern Time. Quick note. In the heart of Cygnus, NASA Fermi reveals the cosmic ray cocoon. Science Daily, November 28, 2011. The constellation Cygnus, now visible in the western sky as twilight deepens after sunlight, hosts one of our galaxy's richest known stellar construction zones. Astronomers viewing the region of visible wavelengths see only hints of this spectacular activity thanks to the veil of nearby dust clouds forming the Great Rift, a dark lane that splits the Milky Way, a faint band of light marking our galaxy's central planet. And good morning, Dr. Will, CBMD PhD. It is Tuesday, November 29, 2011, 3:59 a.m. Eastern Time. Quick note. Located in the vicinity of the second magnitude star Gamma Cygni, the star-forming region was named Cygnus X when it was discovered as a diffuse radio source by surveys in the 1950s. Now a study using data from NASA Fermi Gamma Ray Space Telescope finds that the tumult of star birth and death and Cygnus X-ray has menaced to coral fast-moving particles called cosmic rays. And good morning, Dr. Will, CBMD PhD. This Tuesday, November 29, 2011, 4:01 a.m. Eastern Time. Quick note. Cosmic rays are subatomic particles, mainly photons, that move through space at nearly the speed of light. In their journey across the galaxy, the particles are deflected by magnetic fields, which scramble their paths and make it impossible to backtrack the particles to their sources. And good morning, Dr. Will, CBMD PhD. It is Tuesday, November 29, 2011, 4:02 a.m. Eastern Time. Quick note. Yet when cosmic rays collide with interstellar gas, they produce gamma rays, the most energetic and penetrating form of light, that travel to us straight from the source. By tracing gamma ray signals throughout the galaxy, Fermi's Large Area Telescope, LAT, is helping astronomers understand the sources of cosmic rays and how they are accelerated to such high speeds. In fact, this is one of the mission's key goals. And good morning, Dr. Will, CBMD PhD. It is Tuesday, November 29, 2011, 4:04 a.m. Eastern Time. Quick note. The galaxy's best candidate sites for cosmic ray acceleration are the rapidly expanding shells of ionized gas and magnetic field associated with supernova explosions. For stars, mass is destiny, and the most massive ones, known as types O and B, live fast and die young. And good morning, it's Dr. Will, CBMD PhD. It is Tuesday, November 29, 2011, 4:05 a.m. Eastern Time. Quick note. They are also relatively rare because such extreme stars, with mass of more than 40 times that of our Sun and surface temperatures 8 times hotter, exert tremendous influence on their surroundings, with intense ultraviolet radiation and powerful outflows known as stellar winds. The most massive stars rapidly disperse their natal-class clouds, naturally limiting the number of massive stars in any given region. And good morning, it's Dr. Will, CBMD PhD. This Tuesday, November 29, 2011, 4:09 a.m. Eastern Time. Quick note. Which brings us back to Cygnus X, located about 4,500 light years away. This star factory is believed to contain enough raw material to make 2 million stars like our Sun. Within it are many young star clusters and several sprawling groups of related O and B type stars called OB associations. One called Cygnus OB2 contains 65 O stars, the most massive, luminous, and hottest type, and nearly 500 B stars. And good morning, it's Dr. Will, CBMD PhD. It is Tuesday, November 29, 2011, 4:12 a.m. Eastern Time. Quick note. Astronomers estimate that the association total stellar mass and 30,000 times that of our Sun, making Cygnus OB2 the largest object of its type within 6,500 light years and with ages of less than 5 million years. Few of its most massive stars have lived long enough to exhaust their fuel and explode as supernova. And good morning, it's Dr. Will, CBMD PhD. It is Tuesday, November 29, 2011, 4:15 a.m. Eastern Time. Quick note. Intense light and outflows from the monster stars in Cygnus OB2 and from several other nearby associations and star clusters have excavated vast amounts of gas from their vicinities. The stars reside in cavities filled with hot, thin gas surrounded by ridges of cool, dense gas where stars are now forming. It is within the Hollywood out zones that Fermi's LAT detects intense gamma-ray emission, according to a paper describing the findings that was published in November 25th edition of the Journal of Science. Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 4:17 a.m. Eastern Time. Now, we are seeing young cosmic rays with energies comparable to those produced by the most powerful particles accelerators on Earth. They have just started their galactic voyage, zigzagging away from their accelerator and producing gamma rays when striking gas or straight in the cavities. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 4:18 a.m. Eastern Time. Now, the energy of the gamma ray emission, which is measured up to 100 billion electron volts by the LAT and even higher by ground-based gamma ray detectors, indicate the extreme nature of the accelerated particles. For comparison, the energy of visible light is between 2 and 3 electron volts. The environment holds onto its cosmic rays despite their high energies by entangling them in turbulent magnetic fields created by the combined outflows of the region's numerous high-mass stars. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 4:20 a.m. Eastern Time. Quick note. These shock waves stir the gas and twist and tangle the magnetic field in a cosmic scale, so the young cosmic rays freshly ejected from their accelerators remain trapped in the turmoil until they can leak into quieter interstellar regions, where they can stream more freely. Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 4:21 a.m. Eastern Time. Quick note. The well-known Gamma Cygni supernova remnant, so named for its proximity to the star, also lies within this region. Astronomers estimate its age at about 7,000 years. The Fermi team considers it possible that the supernova remnant spawned the cosmic rays trapped in the Cygnus X-ray cocoon, but they also suggest an alternative scenario where the particles became accelerated through repeated interaction with shock waves produced inside the cocoon by powerful stellar winds. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 4:23 a.m. Eastern Time. Quick note. Whether the particles further gain or lose energy inside the cocoon needs to be investigated, but its existence shows that cosmic ray history is much more eventful than a random walk away from their sources. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 4:26 a.m. Eastern Time. Quick note. Fermi is providing a never-before-seen glimpse of the early life of cosmic rays long before they diffuse into the galaxy at large. Astronomers known as dozen stellar clusters at least as young and rich as Cygnus OB2, including the arcs and quintuplet clusters near the galaxy center. Energetic gamma rays are detected in the vicinity of several of them, so perhaps they also coral cosmic rays in their own high-energy cocoons. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 27, 2011, 4:40 a.m. Eastern Time. Quick note. Marine biodiversity loss due to global warming and predation study predicts. Science Daily, November 28, 2011. The biodiversity loss caused by climate change will result from a combination of rising temperature and predation, and may be more severe than currently predicted, according to a study. University of British Columbia zoologist Christopher Harley. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 4:41 a.m. Eastern Time. Quick note. Harley surveyed the upper and lower temperature limits of barnacles and mussels from the cool west coast of Vancouver Island to the warm shores of the Sand Islands, where water temperature rose from the relatively cool of the 1950s to the much warmer years of 2009 and 2010. The study published in the current issue of the journal examined the response of rocky shore barnacles and mussels to the combined effects of warming and predation by sea stars. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 4:42 a.m. Eastern Time. Quick note. Rocky intertidal communities are ideal test beds for studying the effect of climate warming. Christopher Harley, Associate Professor of Zoology, UBC, authored the study. Many intertidal organisms like mussels already live very close to their thermal tolerance limits, so the impacts can be easily studied. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 4:44 a.m. Eastern Time. Quick note. At cooler sites, mussels and rocky shore barnacles were able to live high on the shore, well beyond the range of their predators. However, as temperature rose, barnacles and mussels were forced to live at lower shore levels, placing them at the same level as predatory sea stars. Daily high temperatures during the summer months have increased by almost 3.5 degrees Celsius in the last 60 years, causing the upper limits of barnacle and mussel habits to relate to 50 centimeters down to the shore. However, the effects of predators and therefore the position of the lower limit have remained constant. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 4:45 a.m. Eastern Time. Quick note: that loss represents 51% of the muzzle bed. Some muzzles have gone extinct locally at 3 of the sites when surveyed. Meanwhile, when pressure from sea star predation was reduced using exclusion cages, the prey species were able to occupy hotter sites where they don't normally occur, and species richness at the sites more than doubled. Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 4:46 a.m. Eastern Time. Quick note: the muzzle bed is kind of like an apartment complex. It provides critical habitat for a lot of little plants and animals. The muzzles make the habitat cooler and wetter, providing an environment for crabs and other small crustacean snails, worms, and seaweed. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 4:46 a.m. Eastern Time. Quick note: these findings provide a comprehensive look at the effects of warming and predation. While many previous studies on how species ranges will change due to warming assume that species will simply shift to stay in their current temperature range. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 4:47 a.m. Eastern Time. Quick note: findings show that the combined effects of warming and predation could lead to a more widespread extinction than are currently predicted, as animals or plants are unable to shift their habitat ranges. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 4:48 a.m. Eastern Time. Quick note: warming is not just having a direct effect on individual species. The study shows that climate change can also alter interactions between species and produce unexpected changes in where species can live, their community structure, and their diversity. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 5:08 a.m. Eastern Time. Quick note: big pest, small genome, two-spotted spider mite genome decoded. Science Daily, November 23, 2011. University of Utah biologist and international research team decoded the genetic blueprint of the two-spotted spider mite, raising hope for new ways to attack the major pests which resist pesticides and destroys crops and ornamental plants worldwide. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 5:10 a.m. Eastern Time. Quick note: big pest, small genome, two-spotted spider mite genome decoded. Science Daily, November 23, 2011. University of Utah biologist international research team decoded the genetic blueprint of the two-spotted spider mite, raising hope for new ways to attack the major pests which resist pesticides and destroys crops and ornamental plants worldwide. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 5:11 a.m. Eastern Time. Quick note: one key thing that makes spider mites unique is they can eat many different plant species. One of the 5 main authors of the study and an assistant professor of biology, University of Utah, these mites often house plants pests, a major cause of people's houseplants turning yellow and getting sick. They also are a major problem for agricultural nurseries and greenhouses and for field crops. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 5:14 a.m. Eastern Time. Quick note: primary targets are tomatoes, peppers, cucumbers, strawberries, corn, soybeans, apples, grapes, and citrus. Clark says the newly studied importance is largely in understanding how animals eat plants, with a long-term goal of developing effective ways to prevent crop damage from mites and insects. If we can identify the biological pathways mites use to feed on plants, we can potentially identify chemical and biological methods to disrupt those pathways and stop the mites from feeding. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 5:15 a.m. Eastern Time. Quick note: the two-spotted spider mite, which is no more than 1 millimeter long, is a major global pest and is predicted to be the growing concern in a warming climate because they multiply extremely fast at high temperatures. 9 degrees Fahrenheit is more, he adds. They do really well in hot and dry climates like Utah. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 5:16 a.m. Eastern Time. Quick note: that the two-spotted spider mite has been found to rapidly develop resistance to multiple types of pesticides, often within a couple of years after a pesticide is introduced. It is resistant to many common pesticides used against insects. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 5:19 a.m. Eastern Time. Quick note: Nature study deciphering the genome of Tetranychus eritrea, the two-spotted spider mite, which has 2 red spots, was conducted by the international research team of 55 scientists from North America, Europe, and South America. And good morning, this is Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 5:21 a.m. Eastern Time. Quick note: genetic blueprint of the two-spotted spider mite. Decoding the spider mite's genome required dozens of scientists with expertise in various gene families. Many studied which genes are expressed or activated, and thus making messenger RNA or mRNA, which in turn is used to make proteins. Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, at 5:21 a.m. Eastern Time. Quick note. The study found that the two-spotted spider mite has 18,414 genes. Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 5:22 a.m. Eastern Time. Quick note. 15,397 genes are expressed or activated to make proteins. Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 5:27 a.m. Eastern Time. Quick note. The spider mite genome contains about 90 megabases. That's 90 million base pairs of DNA letters, A, C, G, and T, which is the smallest genome yet sequenced for any arthropods, which are invertebrate or spineless animals with external skeletons or exoskeleton segmented bodies and appendages with joints. Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 5:28 a.m. Eastern Time. Quick note. Many of the other genomes are enormous, some close to 3 billion bases, or about the size of the human genome, and some up to 7.1 billion bases. Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 5:31 a.m. Eastern Time. Quick note. Arthropods include hexapods, insects, and insect-like animals. Crustacean lobsters, crab, shrimp, barnacles, myriapods, millipedes, centipedes, and chelicerates. Spiders, scorpions, mites, and ticks. Chelicerates are the largest group of animals after insects. The two-spotted spider mite is the first chelicerate to have its genome fully sequenced. Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 5:37 a.m. Eastern Time. Quick note. While there are other species of plant-feeding mites, the researchers chose to sequence the genome of the two-spotted spider mite because of all the spider mites. This is the most widespread because it feeds on so many different plant species. Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 5:38 a.m. Eastern Time. Quick note. The study findings shed light on how the spider mites evolved differently than other arthropods. Compared with other arthropods, the two-spotted spider mite uses a different molting hormone to shed its exoskeleton during growth. Has only 8 HOX genes of orchestrate body plan development, compared with 10 in most other arthropods, and thus has only 2 main body segments instead of 3. There are other cases in which HOX genes were activated differently in different arthropods, but this is an extreme case. Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 5:42 a.m. Eastern Time. Quick note. Big silk is as strong as spider silk, but 185 to 435 times thinner. Spiders spin silk from their abdomen, spider mites from their head region. Spider mites use silk to hide from predators, keep themselves warm, and suspend eggs out of predators' reach. Silk from the mites may prove useful as biodegradable surgical sutures and bandages. It is very thin and very easy to get because you can grow lots of mites on plants. Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 5:49 a.m. Eastern Time. Quick note. A genetic arsenal to attack plant toxins and pesticides. The spider mite genome also revealed the presence of families of genes involved in breaking down toxic compounds, either in plants poisonous to the spider mites or in pesticides. You would imagine that if these mites feed on such a broad number of plant hosts, they would have many genes known to be involved in breaking down toxic compounds, and we found that they did. Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 5:49 a.m. Eastern Time. Quick note. In some specific families of detoxification genes, in the spider mites, the number of genes was about 3 times that seen in other arthropods. Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 5:54 a.m. Eastern Time. Quick note. As part of the study, the scientists took a specific strain of spider mites that normally eat kidney beans and transferred them to tomato and thale cress, Arabidopsis plants. On these new plants, the mites expressed or activated different genes and thus made different detoxification compounds so they could eat the new plant species. Some of those detox genes were previously unknown and thus provide new insights into how mites counteract plant defenses. Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 5:56 a.m. Eastern Time. Quick note. For example, half of the cytochrome P450 family of detoxification gene changed for expression, turned either on or off when the mites were switched to the new plants. That is a bigger change than seen before in any group of animals. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 5:57 a.m. Eastern Time. Quick note. This suggests that these genes are critical for the ability of mites to be pests on many different plants. The spider mite has 39 genes from one drug-resistant gene family, and the proteins they can encode, compared with 9 to 14 in insects and vertebrate animals. That shows how an extended set of genes evolved to help the pest feed on numerous plant species. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 5:59 a.m. Eastern Time. Note: the yet unexplained mystery, the two-spotted spider mite has some genes similar to those in bacteria and fungi. They somehow captured them from other organisms in the environment, and now are using them for their own growth and persistence. They are mostly enzymes involved in changing other small molecules. The hypothesis is these genes may be involved in modifying, detoxifying toxic compounds found in plants. Good evening, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 9:40 p.m. Eastern Time. Quick note: new technique puts chemistry breakthroughs on the fast track. Science Daily, November 28, 2011. Scientists can now take that aha moment to go with a method Princeton University researchers developed and successfully tested to speed up the chances of an unexpected yet groundbreaking chemical discovery. Good evening, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 9:42 p.m. Eastern Time. Quick note: the researchers report this month in the journal Science a technique to accomplish accelerated serendipity by using robotics to perform more than 1,000 chemical reactions a day, with molecules never before combined. In a single day of trials, the Princeton researchers discovered a shortcut for producing pharmaceutical-like compounds that shaves weeks off the traditional process, the researchers report. And good evening, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 9:43 p.m. Eastern Time. Quick note: the basis of the research was to combine new technology with a unique rapid reaction approach that could allow chemists to explore unheard of and potentially important chemical combinations without devoting years to the pursuit. And good evening, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 9:45 p.m. Eastern Time. Quick note: our process is designed specifically for serendipity to occur. The molecules that should be combined are those for which the result is unknown. In our lab, we use this technique to make new findings in a much more routine and rapid fashion, and we show that if you have enough events involved, serendipity won't be rare. In fact, you can enable it to happen on almost a daily basis. And good evening, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 9:47 p.m. Eastern Time. Quick note: the Macmillan's lab technique does more than just expedite the discovery process. The researchers actually developed a unique framework for creating new materials or finding better ways of producing existing ones. And good evening, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 9:50 p.m. Eastern Time. Quick note: usually one takes molecules that one thinks will react and tries to figure out the best ways to achieve that reaction. This team took molecules for which there was no obvious reaction between them and looked for accidental reactivity. This approach could be useful for any field that requires new types of matter or a more efficient means of synthesizing known compounds. And good evening, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 9:52 p.m. Eastern Time. Quick note: illustrating that principle, the Princeton researchers combined two molecules with no history of reacting to generate the type of chemical functionality found in 8 of the world's top 100 pharmaceuticals. The reaction involved a nitrogen-based molecule known as the amine that has a hydrogen and carbon pair, and a circle of atoms stabilized by their bonds known as aromatic ring. Good evening, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 9:56 p.m. Eastern Time. Quick note: the result was a carbon-nitrogen molecule with an aromatic ring, a building block of many amine-based pharmaceuticals. This class of drugs mimics natural amine molecules in the body and includes medications such as antihistamines, decongestants, and antidepressants. In drug development, chemists tweak organic molecules to enhance their ability to bind with and disrupt enzyme in a biological system, which is how pharmaceuticals basically operate. A molecule with an aromatic ring has increased reactivity and makes the tweaking process much easier. By attaching the aromatic ring is a process in itself that typically involves 2 to 3 weeks of successive chemical reactions. And good evening, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 10:02 p.m. Eastern Time. Quick note: we quickly realized that any pharmaceutical research chemist could immediately take these very simple components and via a reaction no one had known about, start assembling molecules with an adjacent aromatic ring rapidly. Instead of having to construct these important molecules, circuitously using different chemistry over a period of days, if not weeks, we can now do it immediately in the space of one chemical reaction in one day. And good evening, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 10:05 p.m. Eastern Time. Quick note. The way these types of molecules, alpha, aryl amines, were produced in this project is highly efficient, and no person could truthfully say that they would have predicted this reaction. This group was able to take a reaction that no one knew was possible and make it practical and useful in a very short time. This really speaks to the power of their overall method. And good evening, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 10:08 p.m. Eastern Time. Quick note. Central to the process is a technique developed in Macmillan's lab and reported in the Science in 2008 to synthesize chemical reactions using a low-power light source, such as a household light bulb, known as photoredox catalyst. The reaction takes place when inorganic catalyst absorbs light. Particles from the light source then pass an electron onto the organic molecules, which creates or synthesizes a new compound. And good evening, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 10:18 p.m. Eastern Time. Quick note. For the latest work, Macmillan and his team carried out this process on the molecules before each reaction cycle. Because the use of photoredox catalysts in organic compound, synthesis is relatively new. It has been typically used by chemists and in industry for processes such as energy storage and hydrogen production. It has not been as thoroughly explored as the more common method of using catalysts derived from metals such as nickel, gold, and copper. Thus, elements with no history of reacting with each other could possibly produce results under this different approach. And good evening, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 10:21 p.m. Eastern Time. Quick note. If one wanted to find new reactions, it would have to be done in a completely new area of chemistry research where the chances of finding something completely unknown are probably higher than continuing in an area that has been studied for the past 50 years. And good evening, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 10:22 p.m. Eastern Time. Quick note. The Princeton research produced numerous new reactions, but new does not necessarily equal interesting or important. Macmillan said, they analyzed and experimented with each new reaction for its potential application, a process that revealed the nitrogen-carbon molecule with the aromatic ring. And good evening, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 10:25 p.m. Eastern Time. Quick note. An important feature of the Princeton researchers' molecule, like an important discovery, is that its application extends beyond the material itself. He and his colleagues have begun mining the very process that created the molecule for indications that other novel reactions can be brought about. If we found this was one really valuable reaction, we wondered what others exist that we just don't know about. And good evening, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 10:27 p.m. Eastern Time. Quick note. Another very valuable aspect of the molecule we created is that once we understood how it happens, it set us up to design other completely new reactions based upon our understanding of what happened initially. Now we are applying similar techniques broadly, finding new reactions continually and determining which ones are important. To us, that really proved the point of why you want serendipitous findings. They present new knowledge, and based upon that new knowledge, you can invent. And good evening, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 10:37 p.m. Eastern Time. Quick note. How bats hear objects in their path. Science Daily, November 28, 2011. By placing real and virtual objects in the flight paths of bats, scientists have shed new light on how echolocation works. And good evening, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 10:37 p.m. Eastern Time. Quick note. The researchers found that it is not the intensity of the echoes that tells the bats the size of an object, but the sonar aperture. That is the spread of angles from which echoes impinge on their ears. And good evening, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 10:39 p.m. Eastern Time. Quick note. Echolocation bats emits calls for orientation. These calls bounce off objects in the bat's environment, carrying information about the object back to the bat. For example, the echoes of large objects are louder than those of small objects. Analyzing echoes when surrounded by a cacophony of calls and echoes from other bats, however, makes this a difficult task for the auditory system. And good evening, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 10:42 p.m. Eastern Time. Quick note. Researchers first wanted to know whether bats are able to use echolocation in such a crowded situation at all. The team filmed the flight paths of hundreds of bats of 13 different species while the bats were emerging from a cave, and then placed a small novel object in the flight paths. And Good evening, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 10:43 p.m. Eastern Time. Quick note. The videos clearly showed curves in the bat's flight paths after we introduced a small novel object. This means that the bats were able to use echolocation in this familiar and crowded situation to detect the object, which measured only 5 times 8 centimeters, and to guide their evasive flight. And good evening, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 10:47 p.m. Eastern Time. Quick note. But how do bats perceive the size of an object from the echoes bouncing off it? To test whether bats use echo intensity, the team used echoes of virtual objects which could be manipulated in size from a loudspeaker. This method records the calls of passing bats and simulates in real time the echoes of objects that are not present physically, just like a projector can show visual images of absent objects. Using this method for the first time with wild bats, the researchers could manipulate a single echo parameter, intensity, and study its effect on the perception of object size. And good evening, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 10:48 p.m. Eastern Time. Quick note. Although the size of the virtual object, and thus its echo intensity, was more than 10 times larger than the small, real object used before, the bats did not show any invasive flight. And good evening, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 10:49 p.m. Eastern Time. Quick note. This result suggests that the virtual object was lacking a crucial feature for object size perception. We think that bats use another echo parameter besides intensity, the sonar aperture, which is a spread of angles of incidence from which echoes impinge on a bat's ear. The sonar aperture directly correlates with the size of real objects, and in contrast to real objects, virtual objects presented from a single loudspeaker lack a wide sonar aperture. And good evening, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 10:52 p.m. Eastern Time. Quick note. The results show that the bats were able to choose the larger objects using a sonar aperture only, independently of echo intensity. This behavior was reflected in the activity of nerve cells that reacted specifically to echoes of a given sonar aperture. And good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, November 29, 2011, 10:54 p.m. Eastern Time. Quick note. Together, these studies have uncovered a novel mechanism for object size perception in bats, which employs the small echoes differences between both ears generated by echoes arriving from different directions. In contrast, our eyes measure object size directly from the two-dimensional retinal image by perceiving the intensity and sonar aperture of object echoes. However, the auditory system has evolved its own solution for the perception of object features, giving bats access to comparable information about objects as we obtain with our eyes. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 23, 2011, 0:58 Eastern Time. Quick note. Smart swarms of bacteria inspire robotics, adaptive decision-making found in bacteria communities. Science Daily, November 17, 2011. Much to humans' chagrin, bacteria have superior survival skills. Their decision-making processes and collective behaviors allows them to thrive and even spread efficiently in difficult environments. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 23, 2011, 1:00 Eastern Time. Quick note. Now researchers at University have developed a computational model that better explains how bacteria move in a swarm, and this model can be applied to human-made technologies, including computers, artificial intelligence, and robotics. Science has discovered how bacteria collectively gathers information about their environment and find an optimal path to grow, even in the most complex terrains. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 23, 2011, 1:02 Eastern Time. Quick note. Studying the principles of bacteria navigation will allow researchers to design a new generation of smart robots that can form intelligent swarms, aid in the development of medical micro-robots used to diagnose or distribute medications in the body, or decode systems used in social networks and through the internet to gather information on consumer behaviors. The research was recently published in PLOS Computational Biology. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 23, 2011, 1:06 a.m. Eastern Time. Quick note. A dash of bacteria's self-confidence. Bacteria aren't the only organisms that travel in swarms. Fish, bees, and birds also exhibit collective navigation. But as simple organisms with less sophisticated receptors, bacteria are not as well equipped to deal with large amounts of information or noise in the complex environments they navigate, such as human tissue. The assumption has been that bacteria would be at a disadvantage compared to other swarming organisms. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 23, 2011, 1:07 a.m. Eastern Time. Quick note. But it's surprising to discover the researchers found that computationally bacteria actually have superior survival tactics, finding food and avoiding harm more easily than swarms such as amoeba or fish. Their secret, a liberal amount of self-confidence. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 23, 2011, 1:08 a.m. Eastern Time. Quick note. Many animal swarms can be harmed by enormous positive feedback, a common side effect of navigating complex terrains. This occurs when a subgroup of the swarm, based on wrong information, leads the entire group in the wrong direction. But bacteria communicate differently through molecular, chemical, and mechanical means, and can avoid this pitfall. Good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 23, 2011, 1:10 a.m. Eastern Time. Quick note. Based on confidence in their own information and decisions, bacteria can adjust their interactions with their peers. When an individual bacterium finds a more beneficial path, it pays less attention to the signals from the other cells. But at other times, upon encountering challenging paths, the individual cell will increase its interaction with the other cells and learn from its peers. Since each of the cells adopts the same strategy, the group as a whole is able to find an optimal trajectory in extremely complex terrain. Good morning, it's Dr. Will, CBMD, PhD. Is Wednesday, November 23, 2011, 1:13 a.m. Eastern Time. Quick note. Benefiting from short-term memory. In the computer model developed by TAU, researchers' bacteria decrease their peers' influence while navigating in a beneficial direction, but listen to each other when they sense they were failing. This is not only a superior way to operate, but a simple one as well. Such a model shows how a swarm can perform optimally with only simple computational abilities and short-term memory. It is also a principle that can be used to design new and more efficient technologies. Good morning, it's Dr. Will, CBMD, PhD. Is Wednesday, November 23, 2011, 1:14 a.m. Eastern Time. Quick note. Robots are often required to navigate complex environments, such as terrains in space, deep in the sea, or the online world, and communicate their findings among themselves. Currently, this is based on complex algorithms and data structures that use a great deal of computer resources. Understanding the secrets of bacteria swarms can provide crucial hints toward the design of new generation robots that are programmed to perform adjustable interactions without taking up a great amount of data or memory. Good morning, it's Dr. Will, CBMD, PhD. Is Wednesday, November 23, 2011, 1:24 a.m. Eastern Time. Quick note. New material could enhance energy, computer lighting technologies. Science Daily, November 16, 2011. Arizona State University researchers have created a new compound crystal material that promises to help produce advances in a range of scientific and technological pursuits. Good morning, it's Dr. Will, CBMD, PhD. This Wednesday, November 23, 2011, 1:25 a.m. Eastern Time. Quick note. The material called erbium chloride silicate can be used to develop the next generations of computers, improve capabilities of internet, increase the efficiency of silicon-based photovoltaic cells to convert sunlight into electrical energy, and enhance the quality of solid-state lighting and sensor technology. Good morning, it's Dr. Will, CBMD, PhD. Is Wednesday, November 23, 2011, 1:28 a.m. Eastern Time. Quick note. The lab's erbium research is supported by the U.S. Army Research Office and U.S. Air Force Office of Scientific Research. Details about the new compound are reported in the Optical Materials Express on the website of the Optical Society of America. Good morning, it's Dr. Will, CBMD, PhD. Is Wednesday, November 23, 2011, 1:29 a.m. Eastern Time. Quick note. The breakthrough involves the first ever synthesis of a new erbium compound in the form of a single crystal nanowire, which has superior properties compared to the erbium compounds and the other forms. Good morning, it's Dr. Will, CBMD, PhD. Is Wednesday, November 23, 2011, 1:29 a.m. Eastern Time. Quick note. Erbium is one of the most important members of the rare Earth family in the periodic table of chemical elements. It emits photons in the wavelength range of 1.5 micrometers, which are used in the optical fibers essential to high-quality performance of the internet and telephones. Good morning, it's Dr. Will, CBMD, PhD. Is Wednesday, November 23, 2011, 1:30 a.m. Eastern Time. Quick note. Erbium is used in doping optical fibers to amplify the signal of internet and telephones and telecommunication systems. Doping is a term used to describe the process of inserting low concentrations of various elements into other substances as a way to alter the electrical and optical properties of the substance to produce desired results. The elements used in such processes are referred to as dopants. Good morning, it's Dr. Will, CBMD, PhD. Is Wednesday, November 23, 2011, 1:31 a.m. Eastern Time. Quick note. Since we could not dope as many erbium atoms in the fiber as we wish, fibers had to be very long to be useful for amplifying an internet signal. This makes integrating internet communications and computing on a chip very difficult. Good morning, it's Dr. Will, CBMD, PhD. Is Wednesday, November 23, 2011, 1:33 a.m. Eastern Time. Quick note. With the new erbium compound, 1,000 times more erbium atoms are contained in the compound. This means many devices can be integrated into the chip-scale system. Thus, the new compound materials containing erbium can be integrated with silicon to combine computing and communication functionalities on the same inexpensive silicon platform to increase the speed of computing and internet operation at the same time. Good morning, it's Dr. Will, CBMD, PhD. Is Wednesday, November 23, 2011, 1:34 a.m. Eastern Time. Quick note. Erbium materials can also be used to increase the energy conversion efficiency of silicon solar cells. Silicone does not disturb solar radiation with wavelengths longer than 1.1 microns, which results in waste of energy, making solar cells less efficient. End. Good morning, it's Dr. Will, CBMD, PhD. Is Wednesday, November 23, 2011, 1:36 a.m. Eastern Time. Quick note. Erbium materials can remedy the situation by converting two or more photons carrying small amounts of energy into one photon that is carrying a larger amount of energy. The single, more powerful photon can then be absorbed by silicone, thus increasing the efficiency of solar cells. Erbium materials also help absorb ultraviolet light from the sun and convert it into photons carrying small amounts of energy, which can then be more efficiently converted into electrically by silicone cells. This color conversion function of turning ultraviolet light into other visible colors of light is also important in generating white light for solid-state lighting devices. And good morning, it's Dr. Will, CBMD, PhD. Is Wednesday, November 23, 2011, 1:38 a.m. Eastern Time. Quick note. While erbium's importance is well recognized, producing erbium material of high quality has been challenging. The standard approach is to introduce erbium as a dopant into various host materials such as silicon oxide, silicone, and many other crystals and glasses. One big problem has been that we have not been able to introduce enough erbium atoms into crystals and glasses without degrading optical quality, because too many of these kinds of dopants would cluster, which lowers the optical quality. And good morning, it's Dr. Will, CBMD, PhD. Is Wednesday, November 23, 2011, 1:41 a.m. Eastern Time. Quick note. What is unique about the new erbium material synthesized by Ning's group is that erbium is no longer randomly introduced as a dopant. Instead, erbium is part of a uniform compound, and the number of erbium atoms is a factor of 1,000 more than the maximum amount that can be introduced in other erbium-doped materials. Increasing the number of erbium atoms provides more optical activity to produce stronger lighting. It also enhances the conversion of different colors of light into white light to produce higher quality solid-state lighting, and enables solar cells to more efficiently convert sunlight into electrical energy. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 23, 2011, 1:48 a.m. Eastern Time. Quick note. In addition, since erbium atoms are organized in a periodic array, they do not cluster in this new compound. The fact that the material has been produced in a high-quality single crystal form makes the optical quality superior to other doped materials. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 23, 2011, 1:49 a.m. Eastern Time. Quick note. Like many scientific discoveries, the synthesis of a new erbium material was made somewhat by accident, similar to what other researchers are doing. We were originally trying to dope erbium into silicone nanowires, but the characteristics demonstrated by the material surprised us. We got a new material. We did not know what it was, and there was no published document that described it. It took us more than a year to finally realize we got a new single crystal material no one else had produced. And good morning, it's Dr. Will, CBMD, PhD. Is Wednesday, November 23, 2011, 1:51 a.m. Eastern Time. Quick note. Ning and his team are now trying to use the new erbium compound for various applications such as increasing silicon solar cell efficiency and making miniaturized optical amplifiers for chip-scale photonic systems for computers and high-speed internet. More importantly, he says there are many things we have yet to learn about what can be achieved with use of the material. Our preliminary studies of its characteristics show it has many amazing properties and superior optical quality. More exciting discoveries are waiting to be made. And good morning, it's Dr. Will, CBMD, PhD. Is Wednesday, November 23, 2011, 2:22 a.m. Eastern Time. Quick note. Form and function new MRI technique to diagnose or rule out Alzheimer's disease. Science Daily, November 16, 2011. On the quest for safe, reliable, and accessible tools to accurately diagnose Alzheimer's disease, researchers from the Perelman School of Medicine, University of Pennsylvania, found a new way of diagnosing and tracking Alzheimer's disease, using an innovative magnetic resonance imaging MRI technique called arterial spin labeling ASL to measure changes in the brain function. The team determined that the ASL MRI test is a promising alternative to current standard, a specific PET scan that requires exposure to small amounts of a radioactive glucose analog and cost approximately 4 times more than an ASL MRI. And good morning, it's Dr. Will, CBMD, PhD. Is Wednesday, November 23, 2011, 2:25 a.m. Eastern Time. Quick note. ASL MRI can be used to measure neurodegenerative changes in a similar way that fluorodeoxide glucose positron emission tomography (FDG) PET scans are currently being used to measure glucose metabolism in the brain. Both tests correlate with cognitive decline in patients with Alzheimer's disease. And good morning, it's Dr. Will, CBMD, PhD. Is Wednesday, November 23, 2011, 2:27 a.m. Eastern Time. Quick note. In brain tissue, regional blood flow is tightly coupled to regional glucose consumption, which is the fuel the brain uses to function. Increases or decreases in brain function are accompanied by changes in both blood flow and glucose metabolism. Radiology senior author on the papers who has worked on ASL MRI for the the past 20 years. We designed ASL MRI to allow cerebral blood flow to be imaged non-invasively and quantitatively using a routine MRI scanner. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 23, 2011, 2:33 a.m. Eastern Time. Quick note. When Alzheimer's disease is suspected, patients typically receive an MRI initially to look for structural changes that could indicate other medical causes, such as stroke or brain tumor, adding about 10 to 20 minutes to the test time. ASL can be incorporated into the routine MRI and capture functional measures to detect Alzheimer's disease upfront, turning a routine clinical test, structural MRI, into both a structural and functional test. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 23, 2011, 2:37 a.m. Eastern Time. Quick note. If ASL MRI were included in the initial diagnostic workup routinely, it would save the time for obtaining an additional PET scan, which we often will order when there is diagnostic uncertainty, and would potentially speed up diagnostics. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 23, 2011, 2:39 a.m. Eastern Time. Quick note. The studies being reported this week show a comparison of ASL MRI and FDG PET in a group of Alzheimer's patients. An age-matched control, cerebral blood flow, and glucose metabolism were measured simultaneously by injecting the PET tracer during the MRI study. The data were then analyzed two different ways. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 23, 2011, 2:42 a.m. Eastern Time. Quick note. In the first study now online, Alzheimer's and Dementia, ASL MRI, FDG, PET images from 13 patients diagnosed with Alzheimer's and 18 age-matched controls were analyzed by visual inspection. Independent blinded review of the two tests by expert nuclear medicine physicians demonstrated similar abilities to rule out sensitivity and diagnose specificity. Alzheimer's neither ASL MRI nor FDG PET showed a clear advantage from quantitative testing. And good morning, it's Dr. Will, CBMD, PhD. Is Wednesday, November 23, 2011, 2:46 a.m. Eastern Time. Quick note. In the second study published in Neurology, the ASL MRI and FDG PET images were compared statistically at each location in the brain by computerized analysis. Data from 15 AD patients were compared to 19 age-matched healthy adults. The patterns of reduction in cerebral blood flow were nearly identical to the patterns of reduced glucose metabolism by FDG PET, both of which differed from the patterns of reduction in gray matter seen in AD. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 23, 2011, 2:57 a.m. Eastern Time. Quick note. Given that ASL MRI is entirely non-invasive, has no radiation exposure, is widely available, and easily incorporated into standard MRI routines, it is potentially more suitable for screening and longitudinal diseases tracking than FDG PET. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 23, 2011, 7:07 a.m. Eastern Time. Quick note. Targeting bacteria gas defense allow for increased efficiency of numerous antibiotics. Science Daily, November 17, 2011. Although scientists have known for centuries that many bacteria produce hydrogen sulfide, H2S, it was thought to be simply a toxic byproduct of cellular activity. Now researchers at NYU School of Medicine have discovered H2S, in fact, plays a major role in protecting bacteria from the effects of numerous different antibiotics. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 23, 2011, 7:08 a.m. Eastern Time. Quick note. In the study, researchers found evidence that H2S acts as a general defense mechanism against oxidative stress, the process through which many antibiotics kill bacteria. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 23, 2011, 7:09 a.m. Eastern Time. Quick note. This information provides the basis for developing new techniques to suppress this universal bacteria defense mechanism and make bacteria more susceptible to antibiotics at lower doses. It also paves the way for reversing antibiotic resistance in human pathogens such as Streptococcus, Pseudomonas, E. coli, and many others. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 23, 2011, 7:11 a.m. Eastern Time. Quick note. Surprisingly little has been known about H2S, biochemistry and physiology in common bacteria. We are excited about the potential impact this research may have on the growing problem of microbial resistance. These findings suggest a conceptually new approach, an adjuvant therapy that targets bacterial gas defense and thus increases the efficiency of many clinically used antibiotics. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 23, 2011, 7:12 a.m. Eastern Time. Quick note. More specifically, the study showed that integrated mechanism of H2S mediated protection against oxidative stress also protects against antibiotics. The research provides direct support for the emerging concept of the pro-oxidative action of many antibiotics. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 23, 2011, 7:13 a.m. Eastern Time. Quick note. In addition, the study demonstrates that bacteria that generate both H2S and nitric oxide NO simultaneously, such as B. anthrax, a causative of anthrax, cannot survive without both gases, even under normal growth conditions. One gas makes up for the lack of the other, and at least one of them is essential. And Good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 23, 2011, 7:15 a.m. Eastern Time. Quick note. In addition, the study demonstrates that bacteria that generate both H2S and nitric oxide, NO, simultaneously, such as B. anthrax, the causative of anthrax, cannot survive without both gases. Even under normal growth conditions, one gas makes up for the lack of the other, and at least one of them is essential. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 23, 2011, 7:18 a.m. Eastern Time. Quick note. In a previous study, his colleagues demonstrated that NO plays a similar role in protecting bacteria from antibiotics. Science, September 9, 2009. However, because NO is present in only a limited number of bacteria while hydrogen sulfide synthesis occurs in essentially all bacteria, the practical implications of this new finding is extremely wide-ranging. And good morning, it's Dr. Will, CBMD, PhD. It is Sunday, November 27, 2011, 1:12 p.m. Eastern Time. Quick note. Smart swarms of bacteria inspire robotics. Adaptive decision-making found in bacteria communities. Science Daily, November 17, 2011. Much to humans' chagrin, bacteria have superior survival skills. Their decision-making process and collective behaviors allows them to thrive and even spread efficiently in difficult environments. And good morning, it's Dr. Will, CBMD, PhD. It is Sunday, November 27, 2011, 1:14 p.m. Eastern Time. Quick note. Now researchers at the university have developed a computational model that better explains how bacteria move in a swarm. And this model can be applied to human-made technologies including computers, artificial intelligence, and robotics. Physics and astronomy from Bar University and Weizmann Institute of Science has discovered how bacteria collectively gather information about their environment and find an optimal path to growth, even in the most complex terrains. And good afternoon, this is Dr. Will, CBMD, PhD. This is Sunday, November 27, 2011, 1:16 p.m. Eastern Time. Quick note. Studying the principles of bacteria navigation will allow researchers to design a new generation of smart robots that can form intelligent swarms, aid in the development of medical micro-robots used to diagnose or distribute medications in the body, or decode systems used in social networks and throughout the internet to gather information on consumer behaviors. The research was recently published in PLOS Computational Biology. And good afternoon, this is Dr. Will, CBMD, PhD. This is Sunday, November 27, 2011, 1:17 p.m. Eastern Time. Quick note. But in the surprising discovery, the researchers found that computational bacteria actually have superior survival tactics, finding food and avoiding harm more easily than swarms such as amoeba or fish. The secret, a liberal amount of self-confidence. And good afternoon, this is Dr. Will, CBMD, PhD. It is Sunday, November 27, 2011, 1:18 p.m. Eastern Time. Quick note. A dash to bacteria self-confidence. Bacteria aren't the only organisms that travel in swarms. Bees and birds also exhibit collective navigation. But as simple organisms with less sophisticated receptors, bacteria are not as well equipped to deal with large amounts of information or noise in the complex environments they navigate, such as human tissue. The assumption has been that bacteria would be at a disadvantage compared to other swarming organisms. And good afternoon, this is Dr. Will, CBMD, PhD. This is Sunday, November 27, 2011, 1:20 p.m. Eastern Time. Quick note. Many animal swarms can be harmed by erroneous positive feedback, a common side effect of navigating complex terrains. This occurs when a subgroup of the swarm, based on wrong information, leads the entire group in the wrong direction. But bacteria communicate differently through molecular, chemical, and mechanical means, and can avoid this pitfall. And good afternoon, this is Dr. Will, CBMD, PhD. This is Sunday, November 27, 2011, 1:22 p.m. Eastern Time. Quick note. Based on confidence in their own information and decisions, bacteria can adjust their interactions with their peers. When an individual bacterium finds a more beneficial path, it pays less attention to the signals from other cells. But at other times, upon encountering challenging paths, the individual cell will increase its interaction with other cells and learn from its peers. Since each of the cells adopts the same strategy, the group as a whole is able to find an optimal trajectory in an extremely complex terrain. And good afternoon, this is Dr. Will, CBMD, PhD. This is Sunday, November 27, 2011, 1:25 p.m. Eastern Time. Quick note. Benefiting from short-term memory. In the computer model developed by TAU researchers, bacteria decrease their peers' influence while navigating in a beneficial direction, but listen to each other when they sense they were failing. This is not only a superior way to operate, but a simple one as well. Such a model shows how a swarm can perform optimally with only simple computational abilities and short-term memory. It is also a principle that can be used to design a new and more efficient technologies. And good afternoon, this is Dr. Will, CBMD, PhD. This is Sunday, November 27, 2011, 1:29 p.m. Eastern Time. Quick note. Robots are often required to navigate complex environments, such as terrains in space deep in the sea, or the online world, and communicate their findings among themselves. Currently, this is based on complex algorithms and data structures that use a great deal of computer resources. Understanding the secrets of bacteria swarms can provide crucial hints towards the design of new generation robots that are programmed to perform adjustable interactions without taking up a great amount of data or memory. And good afternoon, this is Dr. Will, CBMD, PhD. This is Sunday, November 27, 2011, 1:33 p.m. Eastern Time. Quick note. New material can enhance energy, computer, lighting, technologies. Science Daily, November 16, 2011. Arizona State University researchers have created a new compound crystal material that promises to help produce advances in a range of scientific and technological pursuits. And good afternoon, this is Dr. Will, CBMD, PhD. It is Sunday, November 27, 2011, 1:38 p.m. Eastern Time. Quick note. ASU Electrical Engineering Professor. The material called erbium chloride silicate can be used to develop the next generations of computers, improve the capabilities of the internet, increase the efficiency of silicon-based photovoltaic cells to convert sunlight into electrical energy, and enhance the quality of solid-state lighting and sensor technology. And good afternoon, it's Dr. Will, CBMD, PhD. It's Sunday, November 27, 2011, 1:39 p.m. Eastern Time. Quick note. The breakthrough involves the first ever synthesis of a new erbium compound in the form of single crystal nanowire, which has superior properties compared to erbium compounds in other forms. And good afternoon, it's Dr. Will, CBMD, PhD. It is 11/27/2011, 1:41 p.m. Eastern Time. Quick note. Erbium is one of the most important members of the rare earth family in the periodic table of chemical elements. It emits protons in a wavelength range of 1.5 micrometers, which are used in the optical fibers essential to high-quality performance of the internet and telephones. And good afternoon, it's Dr. Will, CBMD, PhD. It is Sunday, November 27, 2011, 1:44 p.m. Eastern Time. Quick note. Erbium is used in doping optical fibers to amplify the signal of the internet and telephones and telecommunication systems. Doping is a term used to describe the process of inserting low concentrations of various elements into other substances as a way to alter the electrical or optical properties of the substances to produce desired results. The elements used in such processes are referred to as dopants. And good afternoon, this is Dr. Will, CBMD, PhD. It's Sunday, November 27, 2011, 1:45 p.m. Eastern Time. Quick note. Since we could not dope as many erbium atoms in a fiber as we wished, fibers had to be very long to be useful for amplifying an internet signal. This makes integrating internet communications and computing on a chip very difficult. And good afternoon, this is Dr. Will, CBMD, PhD. It's Sunday, November 27, 2011, 1:46 p.m. Eastern Time. Quick note. For the new erbium compound, 1,000 times more erbium atoms are contained in the compound. This means many devices can be integrated into a chip-scale system. Thus, the new compound materials containing erbium can be integrated with silicon to combine computing and communication functionalities on the same inexpensive silicon platform to increase the speed of computing and internet operation at the same time. And good afternoon, this is Dr. Will, CBMD, PhD. It is Sunday, November 27, 2011, 1:50 p.m. Eastern Time. Quick note. Erbium materials can also be used to increase the energy conversion efficiency of silicon solar cells. Silicon does not absorb solar radiation with wavelengths longer than 1.1 microns, which results in waste of energy, making solar cells less efficient. Erbium materials can remedy the situation by converting two or more photons carrying small amounts of energy into one photon that is carrying a larger amount of energy. The single, more powerful photon can then be used to absorb by silicon, thus increasing the efficiency of solar cells. And good afternoon, this is Dr. Will, CBMD, PhD. It is Sunday, November 27, 2011, 1:52 p.m. Eastern Time. Quick note. Erbium material also helps absorb ultraviolet light from the sun and convert it into photons carrying small amounts of energy, which can then be more efficiently converted into electricity by silicon cells. This color conversion function of turning ultraviolet light onto other visible colors of light is also important in generating white light or solid-state lighting devices. And good afternoon, this is Dr. Will, CBMD, PhD. It's Sunday, November 27, 2011. While erbium's importance is well recognized, producing erbium material of high quality has been challenging. The standard approach is to introduce erbium as a dopant into various host materials, such as silicon oxide, silicon, and many other crystals and glasses. And good afternoon, this is Dr. Will, CBMD, PhD. It is Sunday, November 27, 2011, 1:56 p.m. Eastern Time. Quick note. While erbium's importance is well recognized, producing erbium material of high quality has been challenging. The standard approach is to introduce erbium as a dopant into various host materials, such as silicon oxide, silicon, and many other crystals and glasses. One big problem has been that we have not been able to introduce enough erbium atoms into crystals and glasses without degrading optical quality, because too many of these kinds of dopants would cluster, which lowers the optical quality. And good afternoon, this is Dr. Will, CBMD, PhD. This is 11/27/2011, 1:57 p.m. Eastern Time. Quick note. What is unique about the new erbium material synthesized by Ning's group is that erbium is no longer randomly introduced as a dopant. Instead, erbium is part of the uniform compound, and the number of erbium atoms is a factor of 1,000 more than the maximum amount that can be introduced in other erbium dopant materials. Thanks. Good afternoon, this is Dr. Will, CBMD, PhD. This is Sunday, November 27, 2011, 1:58 p.m. Eastern Time. Quick note. Increasing the number of erbium atoms provides more optical activity to produce stronger light. It also enhances the conversion of different colors of light into white light to produce higher quality solid state lighting, and enables solar cells to more efficiently convert sunlight into electrical energy. Good afternoon, this is Dr. Will, CBMD, PhD. It's Sunday, November 27, 2011, 1:59 p.m. Eastern Time. Quick note. In addition, since erbium atoms are organized in a periodic array, they do not cluster in this new compound. The fact that the material has been produced in a high-quality single crystal form makes the optical quality superior to other doped materials. Like many scientific discoveries, the synthesis of this new erbium material was made somewhat by accident. Good afternoon, this is Dr. Will, CBMD, PhD. This is Sunday, November 27, 2011, 2:01 p.m. Eastern Time. Quick note. Similar to what other researchers are doing, we were originally trying to dope erbium into silicon nanowires, but the characteristic demonstrated the material surprised us. We got a new material, we did not know what it was, and there was no published document that described it. It took us more than a year to finally realize we got a new single crystal material no one else had produced. Good afternoon, this is Dr. Will, CBMD, PhD. This is Sunday, November 27, 2011, 2:02 p.m. Eastern Time. Quick note. Similar to what other researchers are doing, we were originally trying to dope erbium into silicon nanowires. The characteristic demonstrated the material surprised us. We got a new material, we did not know what it was, and there was no published document that described it. It took us more than a year to finally realize we got a new single crystal material no one else had produced. Good afternoon, it's Dr. Will, CBMD, PhD. It's Sunday, November 27, 2011, 2:02 p.m. Eastern Time. Quick note. Ning and his team are now trying to use the new erbium compound for various applications, such as increasing silicon and solar cell efficiency and making miniaturized optical amplifiers for chip-scale photonic systems for computers and high-speed internet. Good afternoon, it's Dr. Will, CBMD, PhD. It is Sunday, November 27, 2011, 2:03 p.m. Eastern Time. Quick note. Most importantly, there are many things we have yet to learn about what can be achieved with the use of the material. Our preliminary studies of its characteristics show it has many amazing properties and superior optical quality. More exciting discoveries are waiting to be made. Good evening, it's Dr. Will, CBMD, PhD. It's Sunday, November 27, 2011, 9:34 p.m. Eastern Time. Quick note. Form and function new MRI technique to diagnose or rule out Alzheimer's disease. Science Daily, November 16, 2011. On the quest for safe, reliable, and accessible tools to accurately diagnose Alzheimer's disease, researchers from the Hurlin School of Medicine at the University of Pennsylvania found a new way of diagnosing and tracking Alzheimer's disease using an innovative magnetic resonance imaging MRI technique called arterial spin labeling, ASL, to measure changes in brain function. The team determined that the ASL MRI test is a promising alternative to the current standard, a specific PET scan that requires exposure to small amounts of a radioactive glucose analog and costs approximately 4 times more than ASL MRI. And good evening, it's Dr. Will, CBMD, PhD. It is Sunday, November 27, 2011, 9:36 p.m. Eastern Time. Quick note. 2 studies now appear: Alzheimer's and Dementia. The Journal of Alzheimer's Association of Neurology. ASL MRI can be used to measure neurodegenerative changes in a similar way that fluorodeoxide glucose positron emission tomography FDG PET scans are currently being used to measure glucose metabolism in the brain. Both tests correlate with cognitive decline in patients with Alzheimer's disease. And good evening, it's Dr. Will, CBMD, PhD. This is Sunday, November 27, 2011, 9:38 p.m. Eastern Time. Quick note. In brain tissue, regional blood flow is tightly coupled to regional glucose consumption, which is the fuel the brain uses to function. Increases or decreases in brain function are accompanied by changes in both blood flow and glucose metabolism. The papers who has worked on ASL MRI over the past 20 years, we designed ASL MRI to allow cerebral blood flow to be imaged non-invasively and quantitatively using a routine MRI scanner. And good evening, it's Dr. Will, CBMD, PhD. It's Sunday, November 27, 2011, 9:41 p.m. Eastern Time. Quick note. When Alzheimer's disease is suspected, patients typically receive an MRI initially to look for structural changes that could indicate other medical causes. such as stroke or brain tumor, adding about 10 to 20 minutes to the test time. ASL can be incorporated into the routine MRI and capture functional measures to detect Alzheimer's disease up front, turning a routine clinical test structural MRI into both a structural and functional test. And good evening, it's Dr. Will, CBMD PhD. It's Sunday, November 27, 2011, 9:43 p.m. Eastern Time. Quick note. If the ASL MRI were included in the initial diagnostic workup, routinely it would save the time for obtaining an additional PET scan, which we often will order when there is diagnostic uncertainty and would potentially speed up diagnosis. And good evening, it's Dr. Will, CBMD PhD. It's Sunday, November 27, 2011, 9:44 p.m. Eastern Time. Quick note. The studies being reported this week show a comparison of ASL, MRI, and PDG, PET in group of Alzheimer's patients and age matches controls. Cerebral blood flow and glucose metabolism were measured simultaneously by injecting the PET tracer during MRI study. The data were then analyzed two different ways. Good evening, it's Dr. Will, CBMD PhD. It's Sunday, November 27, 2011, 9:45 p.m. Eastern Time. Quick note. In the first study, now online, Alzheimer's and Dementia, ASL, MRI, FDG, PET images from 13 patients diagnosed with Alzheimer's and 18 age-matched controls were analyzed by visual inspection. Independent blinded review of the two tests by expert nuclear medicine physicians demonstrated similar abilities to rule out sensitivity and diagnose specificity. Alzheimer's, neither ASL, MRI, nor FDG, PET showed a clear advantage from quantitative testing. And good evening, it's Dr. Will, CBMD PhD. It's Sunday, November 27, 2011, 9:47 p.m. Eastern Time. Quick note. In the first study, now online, in Alzheimer's and Dementia, ASL, MRI, and FDG, PET images from 13 patients diagnosed with Alzheimer's and 18 age-matched controls were analyzed by visual inspection. Independent blinded review of the two tests by expert nuclear medicine physicians demonstrated similar abilities to rule out sensitivity and diagnose specificity. Alzheimer's, neither ASL, MRI, nor FDG, PET showed a clear advantage from quantitative testing. And good evening, it's Dr. Will, CBMD PhD. It's Sunday, November 27, 2011, 9:49 p.m. Eastern Time. Quick note. In the second study published in Neurology, the ASL, MRI, and FDG, PET images were compared statistically at each location in the brain by computerized analysis. Data from 1580 patients were compared to age 19 matched healthy adults. The patterns of reduction in cerebral blood flow were nearly identical to the patterns of reduced glucose metabolism by FDG, PET, both of which differed from the patterns of reduction in gray matter seen in AD. And good evening, it's Dr. Will, CBMD PhD. It's Sunday, November 27, 2011, 9:51 p.m. Eastern Time. Quick note. Given that ASL MRI is entirely non-invasive, has no radiation exposure that is widely available and easily incorporated into standard MRI routines, it is potentially more suitable for screening and longitudinal disease tracking than FDG, PET, said neurology study authors. And good evening, it's Dr. Will, CBMD PhD. It's Sunday, November 27, 2011, 9:52 p.m. Eastern Time. Quick note. Additional studies will focus on larger sample sizes, including patients with mild cognitive impairment and other kinds of neurodegenerative conditions. And good evening, it's Dr. Will, CBMD PhD. It's Sunday, November 27, 2011, 9:57 p.m. Eastern Time. Quick note. Targeting bacteria gas defenses allow for increased efficiency of numerous antibiotics. Science Daily, November 17, 2011. Although scientists have known for centuries that many bacteria produce hydrogen sulfide, H2S, it was thought to be simply a toxic byproduct of cellular activity. Now researchers at NYU School of Medicine have discovered H2S in fact plays a major role in protecting bacteria from the effects of numerous different antibiotics. And good evening, it's Dr. Will, CBMD PhD. It's Sunday, November 27, 2011, 9:58 p.m. Eastern Time. Quick note. School of Medicine researchers found evidence that H2S acts as a general defense mechanism against oxidative stress, the process through which many antibiotics kill bacteria. And good evening, it's Dr. Will, CBMD PhD. It's Sunday, November 27, 2011, 9:58 p.m. Eastern Time. Quick note. This information provides the basis for developing new techniques to suppress the universal bacteria defense mechanism and make bacteria more susceptible to antibiotics at lower doses. It also paves the way for reversing antibiotic resistance and human pathogens such as Streptococcus, Pseudomonas, E. coli, and many others. And good evening, it's Dr. Will, CBMD PhD. It's Sunday, November 27, 2011, 10:01 p.m. Eastern Time. Quick note. Surprisingly little has been known about H2S biochemistry and physiology in common bacteria. We are excited about the potential impact this research may have on the growing problem of microbial resistance. These findings suggest a conceptually new approach, an adjuvant therapy that targets bacterial gas defenses and thus increases the efficiency of many clinically used antibiotics. And good morning, it's Dr. Will, CBMD PhD. It's Sunday, November 27, 2011, 10:02 p.m. Eastern Time. Quick note. More specifically, the study showed that integrated mechanism of H2S mediated protection against oxidative stress, also protects against antibiotics. The research provides direct support for the emerging concept of the pro-oxidative action of many antibiotics. And good evening, it's Dr. Will, CBMD, PhD. It's Sunday, November 27, 2011, 10:03 p.m. Eastern Time. Quick note. In addition, the study demonstrates that bacteria that generate both H2S and nitric oxide NO simultaneously, such as B. anthrax and causative anthrax, cannot survive without both gases, even under normal growth conditions. One gas makes up for the lack of the other, and at least one of them is essential. And good evening, it's Dr. Will, CBMD, PhD. It's Sunday, November 27, 2011, 10:09 p.m. Eastern Time. Quick note. Block holes can enhance rather than stop light going through. Science Daily, November 22, 2011. Conventional wisdom would say that blocking a hole would prevent light from going through it, but Princeton University engineers have discovered the opposite to be true. A research team has found that placing a metal cap over a small hole in a metal film does not stop the light at all, but rather enhances its transmission. And good evening, it's Dr. Will, CBMD, PhD. It's Sunday, November 27, 2011, 10:11 p.m. Eastern Time. Quick note. In the example of the extraordinary twist of physics that can occur at very small scales, electrical engineer Stefan colleagues made an array of tiny holes in a thin metal film, then blocked each hole with an opaque metal cap. When they shined light into the holes, they found that as much as 70% more light came through when the holes were blocked than when they were open. And good evening, it's Dr. Will, CBMD, PhD. It's Sunday, November 27, 2011, 10:12 p.m. Eastern Time. Quick note. The common wisdom in optics is that if you have a metal film with very small holes and you plug the holes with metal, the light transmission is blocked completely. And good evening, it's Dr. Will, CBMD, PhD. It is Sunday, November 27, 2011, 10:17 p.m. Eastern Time. Quick note. For one, he said, it might require scientists and engineers to rethink techniques they have been using when they want to block all light transmission. In very sensitive optical instruments such as microscopes, telescopes, spectrometers, and other optical detectors. For example, it is common to coat a metal film onto glass with the intention of blocking light. Dust particles, which are unavoidable in metal film deposition, inevitably create tiny holes in the metal film. But these holes have been assumed to be harmless because the dust particles become capped and surrounded by metal, which is thought to block the light completely. And good evening, it's Dr. Will, CBMD, PhD. It's Sunday, November 27, 2011, 10:20 p.m. Eastern Time. Quick note. He explained that in his own field of nanotechnology, light is often used in a technique called photolithography to carve ultra-small patterns in silicone or other materials. Thin metal film patterns on the glass plate serve as a mask directing light through certain locations of the plate and blocking in other locations. Given the new findings, engineers ought to examine whether the mask blocks the light as expected. And good evening, it's Dr. Will, CBMD, PhD. Sunday, November 27, 2011, 10:22 p.m. Eastern Time. Quick note. Conversely, newly discovered blocking technique might be used in situations when a boost in light transmission is desired. In near-field microscopy, for example, scientists view extremely fine details by passing light through a hole as tiny as billionths of a meter in diameter. With the new technique, the amount of light passing through the hole, and thus the amount of information about the object being viewed, can be increased by blocking the hole. And good evening, it's Dr. Will, CBMD, PhD. It's Sunday, November 27, 2011, 10:30 p.m. Eastern Time. Quick note. Stumbled on the phenomenon of enhanced light transmission through a blocked hole, and their research on developing ultrasensitive detectors that sense minute amounts of chemicals, with uses ranging from medical diagnostics to the detection of explosives. These detectors use a thin metal film with an array of holes and metal discs to boost faint signals produced when laser light encounters a molecule, allowing much greater sensitivity in identifying substances. And good evening, it's Dr. Will, CBMD, PhD. Sunday, November 27, 2011, 10:33 p.m. Eastern Time. Quick note. In one of their experimental detectors, the researchers studied transmission of light through an array of tiny holes that were 60 nanometers billionth of a meter in diameter and 200 nanometers apart in the gold film that was 40 nanometers thick. Each tiny hole was capped with a gold disc that was 40% larger than the hole. The disc sat on top of the holes, with a slight gap between the metal surface and the disc. And good evening, it's Dr. Will, CBMD, PhD. It's Sunday, November 27, 2011, 10:36 p.m. Eastern Time. Quick note. On one of their experimental detectors, the researchers studied transmission of light through an array of tiny holes that were 60 nanometers, a billionth of a meter in diameter, and 20 nanometers apart in the gold film that was 40 nanometers thick. Each tiny hole was capped with a gold disc that was 40% larger than the hole. The disc sat on the top of the holes with a slight gap between the metal surface and the disc. And good evening, it's Dr. Will, CBMD, PhD. Sunday, November 27, 2011, 10:40 p.m. Eastern Time. Quick note. The researchers pointed a laser at the underside of the film and tested to see if any of the laser light went through the holes past the caps and could be detected on the other side. To their surprise, they found that the total light transmission was 70% higher with the holes blocked by the metal disc than without blockers. The researchers repeated the same experiment, shining the light in the opposite direction, pointing at the side with the caps and looking for transmitted light under the film, and found the same results. And good evening, it's Dr. Will, CBMD, PhD, Sunday, November 27, 2011, 10:44 p.m. Eastern Time. Quick note. Metal disc acts as a sort of antenna that picks up and radiates electromagnetic waves. In this case, the metal disc pick up light from one side of the hole and radiate it to the opposite side. The waves travel along the surface of the metal and leap from the hole to the cap, or vice versa depending on which way the light is traveling. The research group is continuing to investigate the effect of how it could be applied to enhance the performance of ultra-sensitive detectors. And good evening, it's Dr. Will, CBMD, PhD, Sunday, November 27, 2011, 10:56 p.m. Eastern Time. Quick note. Earth's core deprived of oxygen, Science Daily, November 23, 2011. The composition of Earth's core remains a mystery. Scientists know that the liquid outer core consists mainly of iron, but it is believed that small amounts of some other elements are present as well. Oxygen is the most abundant element in the planet, so it is not unreasonable to expect oxygen might be one of the dominant light elements in the core. However, new research from the team, including the teams, shows that oxygen does not have a major presence in the outer core. This has major implications for our understanding of the period when Earth formed through the accretion of dust and clumps of matter. And good evening, it's Dr. Will, CBMD, PhD, Sunday, November 27, 2011, 10:59 p.m. Eastern Time. Quick note. According to the current models, in addition to large amounts of iron, Earth's liquid outer core contains small amounts of so-called light elements, possibly sulfur, oxygen, silicon, carbon, or hydrogen. In this research, and good evening, it's Dr. Will, CBMD, PhD, Sunday, November 27, 2011, 10:59 p.m. Eastern Time. Quick note. With increasing depth inside Earth, the presence and heat also increase. As a result, materials act differently than they do on the surface. At Earth's center are a liquid outer core and a solid inner core. The light elements are thought to play an important role in deriving the convection of the liquid outer core, which generates Earth's magnetic field. And good evening, it's Dr. Will, CBMD, PhD, Sunday, November 27, 2011, 11:01 p.m. Eastern Time. Quick note. Scientists know the variations in density, speed of sound as a function of depth in the core from seismic observations, but to date it has been difficult to measure these properties in proposed iron alloys at core pressures and temperatures in the laboratory. We can't sample the core directly, so we have to learn about it through improved laboratory experiments combined with modeling and seismic data. And good evening, it's Dr. Will, CBMD, PhD, Sunday, November 27, 2011, 11:06 p.m. Eastern Time. Quick note. High-speed impacts can generate shock waves that raise the temperature and pressure of materials simultaneously, leading to melting of materials at pressures corresponding to those in the outer core. The team carried out shock wave experiments on core materials, mixtures of iron, sulfur, and oxygen. They shocked these materials to a liquid state and to measure their density and speed of sound traveling through them under conditions directly comparable to those of liquid outer core. And good evening, it's Dr. Will, CBMD, PhD, Sunday, November 27, 2011, 11:09 p.m. Eastern Time. Quick note. By comparing their data with observations, they conclude that oxygen cannot be a major light element component of Earth's outer core, because experiments on oxygen-rich materials do not align with geophysical observations. This supports recent models of core differentiation in early Earth under more reduced, less oxidized environments, leading to the core that is poor in oxygen. And good evening, it's Dr. Will, CBMD, PhD, is Sunday, November 27, 2011, 11:12 p.m. Eastern Time. Quick note. The research revealed a powerful way to decipher the identity of the light elements in the core. Further research should focus on the potential presence of elements such as silicon in the outer core. And good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 28, 2011, 1:48 a.m. Eastern Time. Quick note. Scientists turn on fountain of youth in yeast. Science Daily, November 23, 2011. Collaborations between Johns Hopkins and National Taiwan University researchers has successfully manipulated the lifespan of common single-celled yeast organisms by figuring out how to remove and restore protein functions related to yeast aging. And good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 28, 2011, 1:49 a.m. Eastern Time. Quick note. A chemical variation of a fuel gauge enzyme that senses energy in yeast acts like a lifespan clock. It is present in young organisms and progressively diminishes as yeast cell age. And good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 28, 2011, 1:50 a.m. Eastern Time. Quick note. In a report in the September 16 edition of Cell, the scientists described their identification of a new level of regulation of this age-related protein variant, showing that when they remove it, the organism's lifespan is cut short and when they restore it, lifespan is dramatically extended. And good morning, it's Dr. Will, CBMD, PhD. It is Monday, November 28, 2011, 1:51 a.m. Eastern Time. Quick note. In the case of yeast, the discovery reveals molecular components of an aging pathway that appears related to one that regulates longevity and lifespan in humans. And good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 28, 2011, 1:53 a.m. Eastern Time. Quick note. This control of longevity is independent of the type of described previously in yeast, which had to do with calorie restriction. We believe that for the first time we have a biochemical route to youth and aging and has nothing to do with diet. The chemical variation known as acetylation, because it adds an acetyl group to an existing molecule, is a kind of declaration that goes on and off a protein. In this case, the protein SIP2, much like an ornament, can be put on and taken off a Christmas tree. Acetylation can profoundly change protein function in order to help an organism or system adapt quickly to its environment. Until now, acetylation had not been directly implicated in the aging pathway, so this is an all-new role and potential target for prevention or treatment strategies. And good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 28, 2011, 1:56 a.m. Eastern Time. Quick note. The team showed that acetylation of the protein SIP2 affected longevity defined in terms of how many times a yeast cell can divide or replicate lifespan. The normal replicated lifespan in natural yeast is 25. In the yeast genetically modified by researchers to restore the chemical modification, lifespan extended to 38, an increase of about 50%. And good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 28, 2011, 1:58 a.m. Eastern Time. Quick note. The researchers were able to manipulate the yeast lifespan by mutating certain chemical residues to mimic the acetylation and decetylation forms of the protein SIP2. They work with live yeast in a dish, measuring and comparing the lifespans of natural and genetically altered types by removing buds from the yeast every 90 minutes. The average lifespan in normal yeast is about 25 generations, which meant the researchers removed 25 newly budded cells from the mother yeast cell. As yeast cells age, each new generation takes longer to develop, so each round of the experiment lasted 2 to 4 weeks. And good morning, it's Dr. Will, CBMD, PhD. This is Monday, November 28, 2011, 2:33 a.m. Eastern Time. Quick note. Mars Science Laboratory, NASA, launches most capable and robust rover to Red Planet. Science Daily, November 26, 2011. NASA began a historic voyage to Mars, the November 26 launch of Mars Science Laboratory, which carries a car-sized rover named Curiosity. Liftoff from Cap Canaveral Air Force Station abroad, the Atlas V rocket occurred at 10:02 a.m. EST, 7:02 a.m. PST. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 28, 2011, 2:38 a.m. Eastern Time. Quick note. The mission will pioneer precision landing technology and sky crane touchdown to place Curiosity near the foot of a mountain inside Gale crater on August 6, 2012. During a nearly 2-year prime mission after landing, the rover will investigate whether the region has ever offered conditions favorable for microbial life, including the chemical ingredients for life. And good morning, it's Dr. Will, CBND, PhD. It's Monday, November 28, 2011, 2:41 a.m. Eastern Time. Quick note. Curiosity, ambitious science. Goals are among the mission's many differences from earlier Mars rovers. It will use a drill and scoop at the end of its robotic arm to gather soil and powdered samples of rock interiors, then sieve and parcel out these samples into analytical laboratory instruments. Inside the rover, Curiosity carries 10 science instruments with a total mass 15 times as large as the science instrument payloads on the Mars rover Spirit and Opportunity. Some of the tools are the first of their kind on Mars, such as the laser firing instrument for checking the elemental composition of rocks from a distance, an X-ray diffraction instrument for definitive identification of minerals and powdered samples. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 28, 2011, 2:43 a.m. Eastern Time. Quick note. Dreaming takes the sting out of painful memories, research shows. Science Daily, November 23, 2011. They say time heals all wounds, and new research from the University of California, Berkeley indicates that time spent in a dream can help us overcome painful ordeals. And good morning, it's Dr. Will, CBMD, PhD. This is Monday, November 28, 2011, 2:44 a.m. Eastern Time. Quick note. UC Berkeley researchers have found that during the dream phase of sleep, also known as REM sleep, our stress chemistry shuts down and the brain processes emotional experiences and takes the edge off difficult memories. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 28, 2011, 2:45 a.m. Eastern Time. Quick note. The findings offer a compelling explanation for why people post-traumatic stress disorder, PTSD, such as war veterans, have a hard time recovering from distressing experiences and suffering recurring nightmares. They also offer clues into why we dream. Good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 28, 2011, 2:46 a.m. Eastern Time. Quick note. The dream stage of sleep, based on its unique neurochemical composition, provides us with a form of overnight therapy, a soothing balm that removes the sharp edge from the prior day's emotional experience. Good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 28, 2011, 2:49 a.m. Eastern Time. Quick note. For people with PTSD, this overnight therapy may not be working effectively, so when a flashback is triggered by, say, a car backfiring, they relive the whole visceral experience, once again because the emotion has not been properly stripped away from the memory during sleep. Good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 28, 2011, 2:49 a.m. Eastern Time. Quick note. The results offer some of the first insights into the emotional, functional, rapid eye movement, REM sleep, which typically takes up 20% of a healthy human's sleeping hours. Previous brain studies indicate that sleep patterns are disrupted in people with mood disorders such as PTSD and depression. Good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 28, 2011, 2:51 a.m. Eastern Time. Quick note. While humans spend 1/3 of their lives sleeping, there is no scientific consensus on the function of sleep. However, a research team has unlocked many of these mysteries linking sleep to learning, memory, and mood regulation. The latest study shows the importance of the REM sleep state. Good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 28, 2011, 2:52 a.m. Eastern Time. Quick note. During REM sleep, memories are being reactivated, put in perspective and connected and integrated, but in state where stressed neurochemicals are beneficially suppressed. Good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 28, 2011, 2:53 a.m. Eastern Time. Quick note. Those who slept in between images viewing reported a significant decrease in their emotional reaction. The images, in addition to MRI scans, showed a dramatic reduction in reactivity in the amygdala, a part of the brain that processes emotion, allowing the brain rational prefrontal cortex to region control the participants' emotional reactions. Good morning, it's Dr. Will, CBMD, PhD. It is Monday, November 28, 2011, 2:57 a.m. Eastern Time. Quick note. In addition, the researchers recorded the electrical brain activity of the participants while they slept, using electroencephalograms. They found that during REM dream sleep, certain electrical activity patterns decreased, showing that reduced levels of stress, neurochemicals in the brain, soothe emotional reactions to the previous day's experience. Good morning, it's Dr. Will, CBMD, PhD. This is Monday, November 28, 2011, 3:01 a.m. Eastern Time. Quick note. We know that during REM sleep there is a sharp decrease in levels of norepinephrine, a brain chemical association with stress. By reprocessing previous emotional experiences in the neurochemically safe environment of low norepinephrine during REM sleep, we wake up the next day, and those experiences have been softened in their emotional strength. We feel better about them. We feel we can cope. Good morning, it's Dr. Will, CBMD, PhD. This is Monday, November 28, 2011, 3:01 a.m. Eastern Time. Quick note. He was tipped off by the possible beneficial effects of REM sleep on the PTSD patients when a physician at the U.S. Department of Veterans Affairs Hospital in the Seattle area told him of a blood pressure drug that was inadvertently preventing reoccurring nightmares in PTSD patients. Good morning, it's Dr. Will, CBMD, PhD. It is Monday, November 28, 2011, 3:02 a.m. Eastern Time. Quick note. It turns out that the genetic blood pressure drug had a side effect of suppressing norepinephrine in the brain, thereby creating a more stress-free brain during REM, reducing nightmares and promoting a better quality of sleep. This suggests a link between PTSD and REM sleep. Good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 28, 2011, 3:21 a.m. Eastern Time. Quick note. Studying bat skulls, evolutionary biologists discover how species evolve. Science Daily, November 23, 2011. A new study involving bat skulls, bite force measurements, and scat samples collected by an international team of evolutionary biologists is helping to solve a nagging question of evolution. Why some groups of animals develop scores of different species over time while others evolve only a few. Their findings appear in the current issue of Proceedings of the Royal Society B. Biological Sciences. Good morning, it's Dr. Will, CBMD, PhD. This is Monday, November 28, 2011, 3:24 a.m. Eastern Time. Quick note. Research compiled large amounts of data on the diet, bite force, and skull shape in a family of New World bats and took advantage of new statistical techniques to date and document changes in the rate of evolution of these traits and the number of species over time. Good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 28, 2011, 3:26 a.m. Eastern Time. Quick note. They investigated why there are so many more species of New World leaf-nosed bats, nearly 200, while their closest relatives produced only 10 species over the same period of time. Most bats are insect feeders, while the New World leaf-nosed bats eat nectar, fruits, frogs, lizards, and even blood. And good morning, it's Dr. Will, CBMD, PhD. This is Monday, November 28, 2011, 3:28 a.m. Eastern Time. Quick note. One hypothesis is that the evolution of a trait such as head shape that gives access to new resources can lead to the rapid evolution of many new species. Explain connecting changes in body structure to an ecological opportunity requires showing that a significant increase in the number of species occurred in tandem with the appearance of new anatomical traits, and that those traits are associated with enhanced resource use. And good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 28, 2011, 3:30 a.m. Eastern Time. Quick note. If the availability of fruit provided by the ecological opportunity that, in the presence of anatomical innovations that allowed eating the fruit, led to significant increase in birth of new species, then skull morphology should predict both diet and bite force. They found support for these predictions by analyzing thousands of evolutionary trees of more than 150 species measuring over 600 individual bat skulls of 85 species, testing bite force in over 500 individual bats from 39 species in the field, and examining thousands of scat samples to identify the bat diet. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 28, 2011, 3:32 a.m. Eastern Time. Quick note. It can be difficult for evolutionary biologists to demonstrate that traits related to anatomical changes called morphological innovations, such as new skull shape, give certain groups a survival advantage when new food sources such as hard fruits become available. And good morning, it's Dr. Will, CBMD, PhD. This is Monday, November 28, 2011, 3:40 a.m. Eastern Time. Quick note. This study conducted during the International Year of the Bat offers a clear example of how the evolution of new traits, in this case a skull with new shape, allowed animals to use new resources and eventually to rapidly evolve into many new species. We found that when a new ecological niche opened up with an opportunity for bats that could eat hard fruits, they shifted their diet significantly, which in turn led to evolution of new species. And good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 24, 2011, 3:44 a.m. Eastern Time. Quick note. Astronomers take a photograph of the youngest supernova right after its explosion. Science Daily, November 24, 2011. Astronomers have obtained a never-before-achieved radio astronomical photograph of the youngest supernova, 14 days after the explosion of a star in the galaxy. June coordinated telescopes around Europe have taken a photograph of the cosmic explosion in great detail, equivalent to seeing a golf ball on the surface of the moon. And good morning, it's Dr. Will, CBMD, PhD. This is Monday, November 28, 2011, 3:54 a.m. Eastern Time. Quick note. Barely at 23 million light years from Earth, and the constellation can be the scene of one of the most violent phenomena in the universe. Despite its beautiful appearance, the death of a star in the shape of a supernova explosion. Several combined telescopes spread over Spain, Sweden, Germany, and Finland, and the data processing by means of a supercomputer in the Netherlands, enabled to have the capacity of a telescope measuring thousands of kilometers. Thus a really clear image has been obtained, with a detail 100 times greater than that of a space telescope Hubble. This technique, known as radio interferometry, has allowed this team to take a photograph of the supernova SN 2011-DH just some days after its explosion. And good morning, it's Dr. Will, CBMD, PhD. This is Monday, November 28, 2011, 4:02 a.m. Eastern Time. Quick note. This experiment is beating a record. This is the earliest high-resolution image of the supernova explosion. From this photograph, we can define the explosion velocity of the shock wave created in the explosion. The full professor in astronomy and astrophysics from the University of argues that this precision we can look for the previous star on the earlier galaxy photographs, as well as a way up better our future observations. And